Our next presentation is Tessera Therapeutics, and I'd like to welcome Vikram Ranad, Senior Vice President, Corporate Development, to the stage.
Morning. It's a pleasure to introduce you today to Tessera Therapeutics. So at Tessera, our mission is to cure disease by writing in the code of life. And we intend to do this by combining two technologies: gene writing, which is our approach to engineer the genome, and non-viral lipid nanoparticle delivery, which we use to deliver our gene writers to the cell types of interest in the body. We've combined these two to advance a pipeline of in vivo genetic medicines, and our lead program for alpha-1 antitrypsin deficiency, we expect to initiate clinical trials next year. Now, we have about 300 people at Tessera, and that includes internal CMC and internal manufacturing. And we have a GMP-qualified site that we intend to use to supply our clinical trial materials. So let's start with the question, what is gene writing? As everyone in this room knows, CRISPR evolved to break DNA. And it's no surprise it's been remarkably effective at knocking genes out. Tessera was started in 2018 with a simple but powerful premise, which is, what if nature invented a way to write DNA? And we looked to mobile genetic elements, in particular retrotransposons, that do exactly that. They write their sequence into the genome using a mechanism called target prime reverse transcription. And we've developed enzymes that we call gene writers that can edit the genome using that same mechanism, using target prime reverse transcription, or TPRT. And the capability range is very broad, and you see that here on this slide. So we bucket these into three categories: what we call a rewriting on the top, which refers to any small alteration, a short insertion, a short deletion, or a single base pair substitution, and that can be any of the 12 possible single base pair substitutions. In the middle, exon length writing, and we can write exon length sequences and integrate them into the genome with single nucleotide specificity in terms of the site of integration. And at the bottom, whole gene length writing as well. Now, while the intended effect, of course, is at the level of the genomic DNA, we can achieve this by delivering a simple RNA composition of matter. And this schematic gives you a sense of that. So we would intend to deliver an mRNA encoding for the gene writer enzyme. That would be translated, of course, into protein in the cells when delivered to the body. A template RNA, which would be bound by that gene writer protein and dictate where in the genome it should go and what edit it should make. And all of that to be co-formulated in a lipid nanoparticle delivery vehicle. And we think compositions like this have enormous advantages over viral vector-based delivery or ex vivo approaches. So this is our pipeline, and you'll see three categories of programs on it. There are 3 liver monogenic diseases: alpha-1 antitrypsin, PKU, and Wilson's. An in vivo approach for sickle cell disease. And at the bottom, an in vivo and non-viral approach, I should say, for CAR T, where we can durably integrate CAR cassettes into T cells inside the body without any viral delivery. In the interest of time, I won't be able to touch upon our T cell programs today, but we are sharing data on them at ASH in December for those of you who are there. I will walk very briefly through the first 4 programs on this list. And in each of those programs, our intent is to make a true correction of the disease-causing mutation. In the first 3 programs, that would be in hepatocytes, and in sickle cell, of course, in the hematopoietic stem cells. And for each of those programs, we intend to use the rewriting capability I mentioned earlier. And when we do this, we think obsessively, frankly, about 3 parameters. Efficiency, and this is self-evident, the frequency at which we make the intended edit at the target gene. Fidelity, the frequency at which we make unintended edits, but at that same target gene. And then specificity, which is any other edit across the genome at another locus. And what you see here is representative data from one of our programs, in this case our PKU program. And you see certainly very high efficiency. This is in ex vivo hepatoblasts, but also very high fidelity. We have a nearly undetectable level of unintended edits at the gene relevant for PKU, and completely undetectable off-target editing in that specificity column. And I wanted to start here because I think this highlights one of the key differentiators for our approach to genome engineering versus, for example, nuclease or base editing. And the, the great specificity we see here, we think has its molecular basis in the mechanism of action of gene writing in target prime reverse transcription that requires more homology-dependent steps to edit the genome relative to those approaches. So I'll now move into our programs, and I'll start with alpha-1 antitrypsin deficiency. This, as I mentioned, is our lead program. The disease, which is probably familiar to most of you, is one of the most prevalent liver addressable monogenic diseases. And it is caused, in nearly all cases, 95% of patients have the exact same mutation, a single base pair substitution called the PI-Z mutation in the gene encoding alpha-1 antitrypsin. And what happens in these patients is the mutant protein is misfolded, and that misfolded protein ends up aggregating in the endoplasmic reticulum in hepatocytes. And as a result, it cannot be secreted, cannot enter circulation, cannot travel to the lungs and other tissues where it plays an important role. And so you're sort of getting the idea that you have two opposing phenotypes. In the liver, this excessive buildup of mutant protein, a gain-of-function phenotype. And in the lung, inadequate levels of functional protein, a loss-of-function phenotype. And so if you were to simply add exogenous protein, which is currently the only approved category of treatments, plasma-derived augmentation therapy, you may address the lung phenotype, but not the liver phenotype. And conversely, if you were to knock down alpha-1 antitrypsin, for instance with an RNAi, you may address the liver phenotype, but not the lung phenotype. But if you can make a true correction of that PI-Z mutation in hepatocytes, in the cells in which it is produced, you have the opportunity to correct the disease at its source and address both phenotypes. And that is, in fact, our intention. And I'll show you a couple of slides of in vivo data over here. So this is in the PI-Z mouse model. This is a mouse model that has the relevant mutation. It's widely used in the field. And you'll see three panels of data here, DNA on the left, mRNA in the middle, and protein on the right. And this is a single one-time administration of a gene writer delivered as an RNA lipid nanoparticle composition. And on the left, you just see the efficiency. So 92% of the alleles in hepatocytes in these animals end up with a true correction of that PI-Z mutation. In the middle, no surprise then that the mRNA that is transcribed in these animals also harbors that correction. So 93% of the mRNA that we can detect is also corrected. And on the right, you see that we've greatly increased the amount of circulating protein in these animals because so much of it now is wild type and no longer mutant and misfolded. This next slide here, this is non-human primate proof of concept data. And this is, again, one-time delivery of a gene writer. These animals, of course, do not have the mutant, so we're making a surrogate edit at the same gene near the same site of the mutation. And we can correct, again, with very high efficiency. So 79% of the alleles in the hepatocytes are corrected after a one-time administration. And of course, as you would expect, 84% of the mRNA, of course, bears that same edit. We don't have a graph for it, but you can see in the text a very low rate of unintended edits at the target gene, that fidelity rate. So this is our lead program, and we expect to initiate clinical trials for it next year. The data I showed you we believe to be best in category, and we're looking forward to advancing this program. Following fast behind that is our approach for PKU. So PKU is an inborn error of metabolism, and in this case, patients lack the enzyme that metabolizes phenylalanine. And as a result, you have a potentially severe and profound increase in phenylalanine levels that can lead to neurological damage and a variety of clinical impacts. And this is, in fact, such a severe disease that it was added to the newborn screening panel in the United States, in all 50 states, in the 1960s. Our approach here is to make a true correction of the R408W mutation, which is the most prevalent mutation for PKU, and is associated with a classic or severe disease phenotype. And that impacts about 16,000 patients in the US and Europe. And I'll show you just one slide of data over here in the interest of time. This is a transgenic mouse model that harbors that mutation, the R408W mutation. And on the left, you see one-time administration of a gene writer leads to very high efficiency. So 47% of the total liver, which equates to about 78% of the hepatocytes, end up with a correction. And in the graph on the right, you can see a correlation between the amount of editing we achieve, so percent rewriting on the x-axis, and the amount of phenylalanine reduction that is achieved, which is the y-axis. And each dot is an individual mouse. And you can see that as soon as we edit over about 10% of the liver, you have complete normalization of phenylalanine. And that's consistent with some human data as well, liver biopsy data from parents of patients actually with hyperphenylalanemia. So the therapeutically relevant threshold here is 10%, and we're well above that. We have primate data for this program as well, and we're advancing this to the clinic after Alpha-1. I'll briefly talk about our Wilson's disease program. This is a disease where patients lack a copper transport enzyme and are unable to clear copper from the body. So you end up with a toxic accumulation of copper in the liver and other organs that can lead to broad organ damage. And our approach here, like the other programs you saw, is a true correction of the H1069Q mutation, the single most prevalent mutation that impacts about 12,000 patients in the US and Europe. I'll show you again one slide of data. This is a transgenic mouse model that has that H1069Q mutation. And you see one-time administration of a gene writer, the impact on DNA in hepatocytes on the left and mRNA on the right. And these reagents are so potent, we correct essentially all alleles in hepatocytes, as you see on the left, and consequently about 90% of the mRNA in these animals is corrected. The last vignette I'll share with you today is for sickle cell disease. I mean, this is a disease that requires no introduction to this group. I imagine many of you were at the fireside chat next door with Jimmy Olahere. And he's a testament to just how efficacious the ex vivo gene therapies are, like Casgevy, no doubt about that. But you also hear from him just how onerous that patient journey was. In his case, it sounds like 17 weeks in the hospital, a 1-year patient journey, fully myeloablative busulfan conditioning, all the consequences of that. And it's a near certainty that is going to significantly limit how many patients can benefit from those therapies, not only worldwide, but also in the US. So our approach here is a simple IV infusion of our gene writers and lipid nanoparticles that would deliver them to the hematopoietic stem cells in their native niche and make the true correction of the causative mutation, the E6 valine mutation in HBB, without any conditioning or mobilization. And we think we can do this in large part because of our delivery technology. So we developed lipid nanoparticles that can deliver with very high efficiency genetic payloads to these long-term HSCs in their native niche. You see data in humanized mice on the left and primates on the right. These data are with a GFP mRNA, so a transient reporter as a proof of concept. And you see nearly 95% of long-term HSCs in both systems, the humanized mouse and the primate, end up receiving that GFP transcript. I'll just make 2 more points about this data set, which is this was done without any mobilization or conditioning. And the flow gates that we use are very stringent. So we're looking specifically for the long-term HSCs, not the broader population of HSPCs. And we think that's important if you want to have a durable correction of this disease. We've combined this delivery with our gene writers to show that we can edit the HBB gene. We shared these data earlier this year at ASGCT. And so you see a one-time administration gets you 27% editing at HBB in the long-term HSCs. Again, no mobilization or conditioning. And you can add a second dose and increase that figure to about 44%. For context, the bar over here, given that we're making a true correction and not something like a BCL11A knockout, is about 20%. If you correct about 20%, you would have a clinically meaningful benefit. And while I'm showing you 2-dose data here, the intent is certainly a single-dose regimen. We are sharing more advanced data on this program at ASH in December as well, for those of you who were there. So with that, in the interest of time, I will close for today, but thank you very much.